So thank you all for coming on the last day of the conference. I appreciate. So my name is Taichi Suzuki from Arizona State University, and I'm excited to share this preliminary work by Hélène Dion-Phénix, a PhD student in my lab. She's working on this host genetic effects on the gut microbiome. So what factors explain gut microbiome variation? This is a fundamental question in gut microbial ecology because differences in the microbiome can affect the ecology and evolution of animals, right? And this is a classic figure from Ruth Ley back in 2008, characterizing the mammalian microbiome. So each dot is a microbiome representing like 60 different mammals. So if you color code this by diet, it explains a lot of the variation. If you color code this mammalian order, it can also explain a lot of variation. And this is because host phylogeny and dietary types are highly correlated, right? And more studies are confirming this pattern that host phylogeny is one of the best predictors of overall variation in the microbiome, at least in mammals, when you compare divergent species. But we know less about within species variation, so how these host genomic differences can affect population differences in microbiome. So I was interested in this question during my PhD, and we used these wild house mice as a model system. And these are great models because we can use these genetic tools from mouse genetics. And together with the Nachman lab, we collected these population samples across the Americas, shown in orange. And we can also bring these mice back to captivity and do experiments. And we also established these wild-derived inbred lines from 5 locations. And within each location, we have a multiple line as well. And now people can purchase these from Jackson Lab. So to summarize my part of the PhD work, at least in the east coast of North America, we found host genetic distance can predict the microbiome distance better than geographic distance. And once you bring these animals back to captivity, we found that population differences in microbiome persist in this common environment. And when we run these genome-wide association studies using microbiome measurements of the trait, we can find these same genes underlying both mice and human microbiome. So I found this very exciting because it suggests that there can be common genetic mechanisms across mammals to exclude and recruit certain microbes, right? But the problem here is that this is all statistical association, and underlying mechanisms or processes causing this genotype microbe association is unknown. So these are common hypotheses summarized by Kevin Cole that can be underlying these genotype microbe association. So host filtering is one of the popular hypotheses in the field. The idea here is that genetically related animals tend to have more similar immune genes, similar metabolic genes, gut morphology, so on. So they might be able to recruit more similar microbiome, right? But the same pattern can be explained by these other processes, say dispersal limitation of the microbe, right? So genetically related animals tend to live in a similar habitat, prefer a similar diet, then just the passive acquisition of these dispersal limited microbes can also produce this pattern without host filtering. And similar for priority effects, or you can think of it as a maternal effect, where the initial colonization of the microbe can have a very important effect on the assembly process later on, right? Again, this can be happening without host filtering, right? So disentangling these processes are pretty challenging, but our lab is particularly interested in demonstrating this host filtering because this can lead to evolutionarily stable host microbial association across many generations. And there are some indirect evidence of host filtering, and there's pros and cons for each approach. So if you knock out a mouse gene in the mouse genome, like say immune genes, obesity genes, we know it can change the composition. But this approach, we can only test few genes, or it doesn't really necessarily reflect the natural variation in the microbiome or the host genome. And twin studies are an elegant approach comparing monozygotic and dizygotic twins. And the whole assumption is that these different twins will all have exposed to the same environment. But there's also evidence that monozygotic twins tend to be exposed to more similar environments than dizygotic twins. So the environment is not fully controlled. And QTL mapping and GWAS are powerful approach to look for genetic basis of microbiome variation. But often these studies don't control for maternal effects or transmission of microbes from parents to offspring. So we came up with this novel approach to try to test host filtering more directly. So we're exposing these hosts with different genotypes to a same microbial pool. So let me walk you through this. So we're using these wild-derived lines that I just described. So 1 line from Canada, 2 lines from Brazil, 2 lines from New York, and 2 lines from Arizona. And we characterize the microbiome using full-length 16S sequencing using PacBio. So what we did is we took the embryo from these wild-derived lines, and you do a uterine transplant to a common lab line. Okay? So all the mice that was born under the same mom will be exposed to the same microbial pool, right? So the idea is that if we see any differences in the microbiome for the post-transplant animal, it must be due to differences in host genetics. So just to emphasize our approach, this reflects natural genetic variation, environment is controlled, we're looking for genome-wide differences, and we have biological replicates, and importantly, we're controlling for these maternal effects. So let's see some results. So here I'm showing you the difference between lab lines and wild-derived lines before embryo transplant. So the dots in yellow are the lab mice microbiome, and all the triangles are the wild-derived lines. So you can see the separation except the Saratoga B line, and we don't have explanation. But overall, you can see that the mouse lines coming from the same population tend to have more similar microbiomes. And this is not surprising. This is what we know from our previous work. So now let's look at the post-embryonic transplant animal microbiome. And strikingly, we can see that the different genotypes can assemble distinct microbiomes, even from the same microbial pool. So I'll walk you through this too. So on the top, that's the pre-embryonic transplant microbiome, which I just showed in the previous slide. And these are the post-embryonic transplant microbiomes. So they form distinct clusters. And first thing you'll notice is that they kind of cluster by color, suggesting that different genotypes can assemble different microbiomes. And what's most convincing is that these lines from the same population tend to assemble more similar microbiomes, right? So that's a great evidence of host filtering. So if you think about the null hypothesis, if there's no host filtering, we expect all these dots will overlap on top of the yellow dots, right? The mother microbiome. But the fact that they're moving away from the lab mice microbiome and becoming closer to the wild-derived line microbiome is, again, it's suggestive of host filtering. So now let's look at which taxa might be driving these differences. So here I'm just showing bacterial families that relative abundance is more than 1%, and just plotting the top 10 most abundant families, just for simplicity. So, for example, if you look at this brown bar, Bacteroideaceae, you can see they're present in all the lab wild-derived lines, but not in the lab mother, right? And similar thing for this pink taxa, this gray taxa, they're only present in a wild-derived line, but not in the lab line. So let's see what happens after post-embryonic transfer. And they're gone. So we cannot detect these bacteria anymore. And this makes sense, right? Because these animals have only access to the lab mice mother microbiome. So if these taxa are not present in the mother's microbiome, there's no way they can recruit, right? So it's good that there's no contamination, the experiment worked, but the limitation is that they cannot assemble the original microbiome because the microbial pool is different, right? So then let's now look at the taxa that's present in the mother's lab microbiome. So for example, this purple one is Lachnospiraceae, and this is present in all lines. So this seems like one of the core microbial taxa in house mice. And this brown one, Lactobacilliaceae, shows a similar pattern, except this Manaus F line. And this happens to be having the least abundance of Lactobacilliaceae before embryo transplant. After embryo transplant, it's under the detection limit. And I found this very striking because if you imagine these mice are exposed to the microbiome that's 50% Lactobacilliaceae, and once these microbiome go into this mouse gut, it can suppress it less than 1% and enrich these like orange microbes like Akkermansiae, which is very low abundance in the mother, right? So it's a great evidence that different genotypes can enrich and deplete certain taxa, right? And similar pattern for this green one. That's very low abundance in the New York mice, and it even goes lower after post-transplant. So I just received this from my student 2 days ago, so at this point we're just eyeballing the thing, but these are the type of patterns that we're looking for. So in summary, different mouse lines assemble distinct microbiome from the common microbial pool. And mouse lines that's collected from the same population tend to have similar microbiomes. And because they're more genetically related, it's supporting this hypothesis. So it's a strong evidence of host filtering, and there's no evidence of dispersal limitation in this study because all microbes had equal opportunities to colonize the mice. And also there's no evidence of priority effects, right? Because if there's a strong priority effects, we think, we'll predict everything will look the same, right? We'll have the same microbiome. So for the next steps, so we want to ask if we can predict the wild-caught microbiome or pre-transplant microbiome from these post-transplant microbiome data. And also, we're using this full-length 16S sequencing, so we're interested in the strain variation. So we want to ask if genetically related hosts may have this more genetically similar microbial strains. And one undergraduate student is working on this third question by using these previously known genotype microbe associations, and can we see this pattern in our post-transplant mice? So for that, thanks for listening, and I'm happy to take any questions. Talk a little too fast, but any questions you can. So I do have to catch a plane right after this, but please feel free to email me if you have any questions. And yeah, thank you.
So at the end, you just mentioned this full-length 16S sequencing. How do you plan to do that?
Yeah, so it's a full length. It's like 1,500 base pair. We'll try to align it and see if we can make a phylogeny and try.
Plan to account for the error rate. So I'm assuming it's on nanopore or back.
Correct. We have a PacBio representative in the back, but yes. So I guess like one nucleotide error rate will have more effect, right? Because small. But there are papers that did look at strain variation using PacBio 16S. So there's some hope, but we haven't tried it yet. But yeah, good point. Thank you. Yeah, thank you.
Okay. Hi, everyone. My name is Hélène Dion-Phénix, and I'm a PhD student at UQAM here in Montreal. And today I will present you the results of a collaborative project on the consequences of snake predation on nesting site selection in blue tits. Small passerines live only few years and reproduce typically once a year, so each of their reproductive events contributes strongly to their lifetime reproductive success. Thus, those birds should adapt to avoid nest failure. And we talk about nest failure when there's the loss of all the eggs or all the nestlings from a brood, and it can be caused by predation, parasitism, abandonment by the parents, or adverse weather. To be able to adapt, the bird the risk of failure must be non-randomly distributed in space. So here, for example, in this context, okay, you can't see anyway, in this context, it's possible for a bird to avoid risky sites and select less risky sites, while in this context, where the risk is randomly distributed, it's impossible for birds to avoid the risky sites. To be able to adapt, the parents must also be able to identify and avoid risky sites or adjust to past experiences. So for instance, if 1 year the bird experienced a nest failure, the year after it could decide to select another nesting site. We can think that some individuals may be better than others at assessing the risk. For example, due to experience, aging could increase the ability to assess the risk, and slow explorers should get more precise information from their environment and thus be better at assessing the risk than fast explorers. To study the consequences of predation and more generally of nest failure, we studied 3 populations of Corsican blue tits that nest in artificial nest boxes and that experience nest failure. These birds live in contrasting habitats and present many morphological, physiological, and life history differences. The population of D. muro on your left side is dominated by deciduous oaks, while the population of E. muro and E. perio is dominated by evergreen oaks. The birds from D. muro live in richer habitat, lay earlier, lay more eggs each year, and have a faster pace of life than birds that nest in evergreen habitats. This contrast must be taken into consideration for all studies on this population, but it also represents an opportunity to broaden the context of our study. So in our study system, we can tear apart 2 main causes of nest failure. So when between 2 subsequent visits we observe the loss of the old clutch, we conclude that there has been predation, which is mainly caused by snakes in our population, because snakes is the only predators in Corsica that can enter the nest box by the hole and have access to the clutch. On the picture, you can see a snake that just ate all the nestlings. The other main cause of nest failure is abandonment. And when between 2 visits we observe the death of all nestlings or the interruption of the brooding, we conclude that the parents abandoned the nest. For this study, we were interested in the spatial and temporal patterns in the risk of predation in our natural populations. And we also wanted to know if the probability of changing a nesting site the following year depend on nest failure, and if this probability was affected by individual threats of the birds. So we use a long-term program data set to answer these questions. This program started in 1976, and this allowed us to have enough failure events, even though they are rare, as you can see by the numbers. And for the rest of the presentation, I won't go into deep statistical details, but all following results are, for all the results, we use binomial mixed models to test our predictions. So for the first part of the results, I will show you the result concerning spatial and temporal patterns in the probability of predation. We were expecting the probability of predation to vary between the 3 populations, also between zones that present different habitat characteristics, and between nest boxes, and it's indeed what we found. Our results indicate that the probability of predation is higher in the 2 evergreen populations compared to the deciduous one. And we see that some zones and some nest boxes are more risky or have a probability of predation higher than others. So here, for example, the zone that is in the center has a lower risk of predation than the 2 other zones. So this is one map from one of our sites. And the color represents the predation risk. The size of the dot represents the occupation rate of the nest boxes. And there's a tendency that riskier nest boxes are less often occupied than non-risky ones, although this tendency is not significant. But these results indicate that the probability of predation is non-random, and thus that there is a potential for birds to predict and avoid nest failure. Now, regarding temporal patterns, our results show an increase in the probability of predation with laying date. And we know that snake activity increases with temperature with a peak around May, and thus it may be why it's riskier to lay later in the spring. We also looked at temporal variation through the years and found that the probability of predation increase in the E. muro population, stay relatively stable in E. perio, and decrease in D. muro. So we know that in our population, the mean laying date varies between the population, but also that this laying date is changing through times in our study systems. Thus, the difference between the difference in the relations may be caused by variation in the synchronicity between snake activity and bird reproduction that differ between our populations and through time. Now for the second part, I will present you our result on the response of birds to nest failure. Our results indicate that the probability of changing nesting site increased the year following nest failure, but only for females and not for males. So you can see that after a year of success, the female has about 70% of probability to change nesting site, while this probability increased to about 90% after a failure. While for male, it stays constant to about 60%, although you can see that the variance increase after failure. So what this result indicates indirectly is that failure increases divorces because the females change nesting sites but not the males. And an older study on our population suggests that female is the sex that decide to divorce, and that divorced females tend to move larger away from the nest and improve on average their breeding site quality, while males tend to have a stronger site fidelity. Our results also show that the probability of changing nesting site decrease with age the year following nest failure. We were expecting a positive relationship because we thought that with age, bird will gain experience and would be better at assessing the risk, but it doesn't seem to be the case. In our populations, we know that high-quality nesting sites are occupied by older individuals. So thus, it's possible that the decision to stay in high-quality nesting site exceed the decision of changing after a nest failure. And indeed, we do see that the probability of changing nesting site decrease with the occupation rate, which is linked to site quality. We were also interested in the effect of exploration speed assessed by open field tests on the response to failure, and we were expecting slow explorers to respond more than fast explorers to failure. But our results indicate that it seems that the probability of changing nesting site after a failure did not change with exploration speed. Thus, it seems that adjusting to nest failure is a conserved threat across individuals. We also wanted to compare the response to failure between our populations, and we were expecting a stronger response in the riskier population. We do see that the probability of changing is higher in the population where the risk is higher, so in the evergreen habitats, but the response to failure do not vary between the three populations. So it's possible that because of the importance of nest failure on lifetime reproductive success, the birds may all be adapted to avoid it even in populations that present a relatively low risk of predation. So in conclusion, we found that the probability of predation is non-randomly distributed in space and time. And our results also suggest that the parent blue tits can adjust their nesting site selection based on past experiences. And with our results taken all together, we think that, after a failure, the parents will divorce, females will change nesting site to try to increase their site quality and probably their mate quality as well. Young male may try to do the same, but it may be harder for males to find quality nesting site because of the competition for the access to good territories. And the older males that were in average and already in good territories regarding other aspects than predation risk, like food availability, for instance, will probably stay to the same nesting site even after nest failure. Our results also indicate that adjusting to nest failure seems to be a conserved behavior across populations that present different risk of predation and between individuals with different exploration profiles. And finally, we want to highlight that long-term programs allow the study of rare but meaningful ecological events like nest failure. And for the future of this study, we want to determine which habitat characteristics are associated with high risk of failure, how parents are able to identify risky sites. Is it by the identification of habitat characteristics, or is it by social learning, for instance? And finally, we are interested in the fitness consequences of changing or not following a failure event. Thank you for listening, and I think we have time for questions.
Is all in nest boxes. I'm wondering how much of it has been validated in natural nesting cavities, where there might be different predators and a different history of selection.
Yeah, it's a good question. We have another population that we follow in Montpellier, and they do have other predators. So it might be something that we want to look at in the future. But in Corsica, it's quite particular that there's almost only snake that can access the nestlings. Another question? Thank you.
Hi everyone, I'm Brooke. I'm a PhD student in the Genu Wolfson Lab at UMass Lowell. And I'm going to tell you a little bit about my research investigating how microbiomes contribute to resiliency in stony coral tissue loss disease. So this disease affects corals within the order of Scleractinia. So these are stony reef-building corals, and it's a very large group, as you can see from some of the pictures of the species included. It's really diverse. They have varying morphologies and lifestyles, but what they have in common is that they're ecosystem engineers. So these are the corals that are building the physical reef habitat, and so that obviously makes them really important to coral reef ecosystems. Stony coral tissue loss disease, or SCTLD as we call it, emerged in 2014 in Florida and has since been spreading throughout the Caribbean. And so it can have really high mortality rates, up to 90% in some cases. So it's really been devastating some of these reefs. So this is a picture of what the disease looks like. It's characterized by this white lesion that spreads across the colony and leaves behind dead skeletons. So that white part is the dead skeleton that's left, and the brown is the living tissue. So we know that this disease is infectious, but we do not know what the pathogen is. There's still some mixed evidence for both viral or bacterial, or it could even be a multi-pathogen disease. And what's unique about this disease is that it affects over 20 species of Scleractinia, and this is pretty rare for coral diseases. Most of the time, they're only affecting a few species. And the ones that are affected, they show approximately the same disease signs, as you can see from some of these pictures here, but they do exhibit differences in susceptibility. So some have much higher infection and mortality rates than others. So when we consider why some of these corals are more resistant or more susceptible than others, it's important that we consider the coral holistically. And so if you don't know, I just want to note here that an individual, or a colony as we call it, is actually made up of many individual animals. So it could be, so the polyps are like the individual animals, that's what the circles or ellipticals are. And so a colony could have tens, hundreds, thousands of genetically identical polyps all living together and functioning together. So corals also function as a holobiont. So if you're not familiar with that term, it's the same as like a metaorganism. It's this idea that a host, in this case the coral animal, functions together with all of its inhabiting microorganisms. So corals have a very well-known, well-established symbiotic relationship with algae, and so these are endophotosymbionts in the family Symbiodinaceae, so these live within the coral tissue and provide the majority of the coral's nutrient requirements. So most Scleractinia cannot live without them, so these are a really important part of their microbiome. But on top of that, they have a very diverse and complex microbiome consisting of bacteria, viruses, fungi, and archaea. And so all of these provide essential functions to the hosts, and that could include nutrient cycling or immune system support. And so our question that we're asking here is, how do these coral species evolve this distinct disease resistance, specifically within stony coral tissue loss disease? And so our group looks at the entire holobiont, so that includes the host system, the symbiont community, and then I specifically am looking at the microbiome. So how do we do this? We have our study site in Belize, where in 2019 the reefs were healthy, meaning the disease had not reached them yet. And so we set up some sites tagged and sampled corals of about 5 target species that span that susceptibility range. And then at some point between 2019 and 2022, there was an SCTLD outbreak, in which case we returned and resampled our tagged colonies. And then since then, we've been processing those samples. We're using shotgun sequencing, and I'm working on some bioinformatic analyses of those sequences. And I'll talk a little bit more in detail about that in a minute. So first, just looking at how the corals were affected on a demographic scale. So this graph is showing you the change in health status over time. So each bar is a coral species, and then on the y-axis, you have the number of colonies that reflect the condition. And so in 2019, we see here that all of them were healthy. And then by 2022, we found that about 60% of all of our tagged colonies were affected by SCTLD. And so this again is showing that range in susceptibility depending on the host coral species. And so just to point out a few of these, Meandrina is known to be one of the most susceptible, and so we found that here as well. All of our Meandrina colonies were dead by 2022. And then the ones we're going to focus on today they're both in the more intermediate range, Pister and MCAV, but Pister does have a higher mortality rate. So since we've been tracking these colonies through time, before, during, and after disease, we know which colonies were susceptible or resistant to the disease. So we're comparing the metagenomes of these susceptible and resistant colonies to try to identify any biomarkers of resistance by like microbial taxa. So like I said, our 2 species that we're going to be talking about today is Pseudodiploria strigosa, or Pister, is our highly susceptible example, and Montassia cavernosa, or MCAV, is our intermediate. And so we call a colony susceptible if it eventually got the disease versus resistant if it never showed disease signs. And so we have a mix of these colonies within each species so we can compare between the susceptible and resistant within a host species and between the 2 host species. Okay, so like I said, we collect our coral tissue samples. We're extracting the DNA, performing microbiome enrichments, and then using shotgun sequencing to collect short reads of all of the holobiont DNA. And then during my QC and alignment process, I'm filtering out the coral host sequences as well as the algal sequences, and so I'm just focusing on the microbial community. And so right now I'm in the midst of doing some metagenomic analyses, so that includes taxonomic profiling, functional profiling, and biomarker identification. So who's there, what are they doing, and how does this differ between the susceptible and resistant colonies? And so today I'm just going to show you my initial results in our taxonomy and our DSeq, so that's comparing the different types of colonies. So first we just look at the microbiome composition as a whole. And when we did that, we found that species was a more significant driver of community composition than disease state. So this NMDS plot is going to show you the differences of the microbiome. So this is using results of beta diversity, Bray-Curtis dissimilarity. So these points, the closer they are together, that means their microbiome composition is more similar versus ones that are farther apart have less similar microbiomes. So this clustering by color shows us that the samples within the same species have more similar microbiomes. And just to point out our different health statuses that are included in this plot, we have our healthy samples, which are taken from a colony with no disease signs. Disease tissue is the apparently healthy or the living tissue on a disease colony, and disease margin is a sample taken right on that lesion line. And so I was a little surprised that we didn't see more similarities based on disease state, especially because some previous studies have seen dysbiosis as a result of this disease. But this, again, is supporting that these species do have very species-specific microbiomes. Okay, and then we can compare the taxonomic groups between the susceptible and resistant colonies. So this is just looking at MCAV, our intermediate species. So this is the result of DSeq. So again, comparing the taxonomy between the 2, and the ones listed here all were significant. So on our x-axis, we have the microbial species, and then on the y-axis is the log-fold change. So if it has a positive change, that means it was more abundant in susceptible colonies versus a negative log-fold change was more abundant in resistant colonies. So in our MCAV, we found 11 taxa that were significantly more abundant in resistant colonies. So this is pretty exciting because these might be some biomarkers of resistance. And I have not yet had a chance to dive into each of these, but I do want to point out Endozoica monas. This strain has been continually popping up in coral microbiome studies, particularly with healthy colonies. And it actually has been associated with resistance to white band disease, which is another coral disease that shows some very similar disease signs but affects different coral species. So it's pretty cool that we're seeing that consistency here. And then when we do the same analysis with our more susceptible species, Pister, we actually found no taxa that were significant to resistant colonies. So instead, we found one taxa that was significantly more abundant in our susceptible colonies. And this is a Vibrio strain. And this is also pretty interesting because Vibrio, different Vibrio species have been found in association to a lot of coral diseases, SCTLD included, not this strain, but other Vibrios. And they're kind of known as opportunistic pathogens, so it could be that these Vibrios are commensally occurring in the Pister microbiomes, and then as soon as they get stressed or get that initial pathogen infection, they're acting as opportunistic pathogens and exacerbating the disease. So when we compare these results between MCAV, our intermediate species, and Pister, our highly susceptible species, we do find that these species-specific microbiomes might be contributing to disease resistance. So kind of reiterating, Pister might be lacking those beneficial microbes that were significant to the resistant colonies within MCAV, or they might have too much of these opportunistic bacteria, including Vibrio, that are making them more susceptible. Or, and more likely, it could be a combination of these factors in addition to other aspects of the holobiont that we haven't really looked at yet, but including the differences in host genetics and differences in symbiont community. So to summarize, here we have further supported that coral species are differentially affected by SCTLD. And the results that we have here are consistent with what's been seen across the Caribbean so far. And we have some preliminary results that suggest that the differences in susceptibility might be due to these species-specific microbiomes. We've also identified some potential biomarkers of resistance, including Endozoicomonas. And so going forward, I'm really excited to dive a little bit deeper into those biomarkers. I want to expand my bioinformatic analyses to explore their genetic potential and try to identify exactly how they might be contributing to resistance, and then see if they are present in other resistant species. So of course, I'm going to repeat this analysis to our other susceptible species that I didn't get to touch on today. So with that, I would like to thank my lab for all of the hard work that's gone into this project so far, our funding sources, and thank you all for listening, and I'm happy to take any questions. Yeah. Good question. There was only one, and it was, I don't remember what it is right now off the top of my head. But I think it was included in one of the MCAVs, so I feel like it was just being driven by the MCAV results. Yeah. But yeah, it was a little bit different when I combined them. Yeah, and just to be curious, considering you're also looking at additional corals. Yeah, combining them all and just seeing, yeah, definitely. Thank you. Sorry, non-infected, infected, and what? Recovered. Meaning like some yes, definitely. I haven't gotten there yet, but I definitely want to do that because, yeah, some of them did get the disease and then they stopped showing those lesion progression. So we do have samples of that. Thank you. Yeah, it's definitely a good idea.
Is there something about like their mucus composition or something on the outside of the corals that's fostering those two different bacterial communities that you find?
Probably. So the samples that we take includes mucus and tissue, and probably a little bit of skeleton. Yeah, unfortunately, we don't have the ability right now to like separate it out. But I think some other studies have looked at the differences in location, and I think mucus is pretty important to bacterial. Yeah. right, thank you guys.
Hi, my name is Danny Morales. I'm a PhD student in Florida International University in the Matt DeGeneiro Lab. And today I'm going to be talking to you about some of my work for my dissertation, where we're trying to understand how Aedes aegypti, the dengue-carrying mosquito, has actually co-opted a family of long terminal repeat retrotransposons and developed a sort of CRISPR-like antiviral immune system unique to invertebrates in this case. So a little bit of background, I work with the Aedes genus of mosquitoes. Aedes is responsible for carrying many of the major viral diseases that we associate with mosquitoes, including dengue, Zika, chikungunya, yellow fever, and so on. There are two species. We have Aedes aegypti, Aedes albopictus. Both of these species have really expanded in the past few years and colonized most of the planet, and we're seeing increasing threat of arboviral disease throughout time. With that, normally when we look at mosquito-virus interactions, it tends to be a very one-sided relationship where we're really just considering the mosquito's innate immune system and how that is affecting the virus and how the virus is affecting the mosquito. It is very one-sided sort of conversation, but in reality, that's not what's happening in the mosquito. The mosquito is a complex individual. There are many within-host conflicts that are happening, including conflicts between the mosquito and its transposable element landscape, where the mosquito is trying to fight off genome size expansion by way of P-mediated transposable element silencing, as well as there is a level of competition for resources between transposable elements and viruses within their replication cycle in the cytoplasm. However, recently there's been a little bit of work that's come out of the University of San Francisco where we have been able to see that these mosquitoes have been able to co-opt long terminal repeat retrotransposons, or LTRs, in order to capture viral information and integrate it into loci within their genome. This is very similar to sort of the original CRISPR-like, or the original CRISPR loci that we see in bacteria, where they capture phage information and integrate it into spacers. So what will usually happen is if an RNA virus is replicating in the same cell that a retrotransposon is replicating, that reverse transcriptase that the LTR is encoding may capture the viral information, integrate it into the LTR transcript, and it'll eventually get integrated right into that genome. These integrated sequences are known as endogenized viral elements, which can then produce piRNAs, which are a class of small RNAs that bind to the protein Pee-wee, which will then bind, like I said, to the Pee-wee protein in the nucleus or the cytoplasm, and they can usually degrade transposable elements. That is their original function. However, in Aedes mosquitoes, we've seen that they are also able to suppress viral replication throughout the viral life cycle. And so with that, there's a bit of an updated figure of what this sort of really should look like. There is a level of cooperation that we are seeing between the mosquito and its transposable element landscape, where these transposable elements are serving as the nucleus or the capturing process of viral sequences, and the mosquito has sort of relaxed its anti-TE defenses. Of course, there's an effect that comes with that. You are having an expanding genome size, which I'll talk to you a little bit later. But you do get the very important added benefit that you now have a secondary adaptive immunity against these viral pathogens. And one of the exciting aspects of this is that it is heritable, so it can be transmitted down through your generations. Now, this system, while it's still in its infancy in terms of what we understand about it, we have noticed that it is an extremely biased system. There are certain viral families that are biased significantly more than others, particularly rhabdoviruses, which are generally plant-borne viruses. There's a lot of theory behind whether there is a transmission cycle between plants and mosquitoes during nectar seeking, as well as flaviviruses, which have all of our favorites, dengue, Zika, yellow fever, and so on. However, one of the things that when this paper first came out of UCSF that I noticed is that there are certain viral families that are missing. One of them, for example, is Toga viridae, which contains the chikungunya virus. This was a huge issue around 2016, 2018, particularly in Brazil, and has continued to ravage throughout Central and South America. And so there are these two explanations, whether there's just more of certain viruses in the wild, so they're just getting integrated more often, or whether there's an actual preference. And that's something that I'll talk to you guys later on today. So we have this system where Peewee has found a sort of new role in Aedes aegypti. It is targeting these viruses, and it's suppressing both transient viruses as well as persistent viruses that are maintained within that population throughout their entire life cycle. And so when we saw this, we had the hypothesis that there was a sort of molecular domestication of the LTR itself that was overcoming some of the fitness hits of an expanding genome size by allowing them to generate this adaptive immunity. So we wanted to look at expanding genome size as our first question. And what we saw is that the genome sizes of many of these mosquitoes have expanded rapidly in the past 100 million years. So this is the phylogeny of Culicidae. We generated it using around 1,600 Busco genes that are shared across all of these individuals. And we find that the Anopheles clade, which is this clade up here, is maintaining the conserved genome size of about 100, 120 megabase long genomes in comparison to the non-Anopheles clade, which is this lower clade down here. When we look into that lower clade, we can see over here in the bottom, we have Aedes aegypti, Aedes albopictus, which have had massive genome size explosions. Aedes aegypti has a genome size of about 1.4 gigabases, while Aedes albopictus has a genome size of anywhere between 1.8 to 1.9, depending on what resource you look at. We also see Armagirus, which is a very close cousin of the Aedes clade, has a very similar huge genome size. So we see this massive radiation. However, we also see Culex. Culex is another genus of mosquitoes. They tend to actually cohabitate very similar niches, and Culex has maintained the ancestral genome size. So this was something that was curious to us. And so we wanted to know, are there more of these Peewee proteins that sort of suppress genome size expansion? What's going on here? And so we looked at the expansion of Peewee orthologs in mosquitoes. And mosquitoes in general have actually had a massive expansion of this Peewee arsenal, particularly this lower clade down here. The Anopheles clade has maintained the ancestral 2 to 3 Peewee setup, similar to what we see with our outgroups, which are non-biting midges. However, when we split off from Anopheles, there is a rapid series of radiation events where we have, duplication events where we have increasing numbers of Peewee orthologs from 3 to 4 to 6 and eventually 7 in the case of Aedes aegypti. And one of the interesting things that we find is when we look at the Aedes aegypti area, and it'll move in a second, it's a very slow PowerPoint, the expansion of the genome size actually happened after the expansion or our predicted expansion of Peewee. So it was indicating that Peewee isn't really doing its job correctly. It's not suppressing viral genome size expansion. It's not doing what it's supposed to do. So we wanted to know, are TEs expanding? You know, this entire system is based on TEs, and particularly RTEs that are able to capture viral sequences expanding and driving this genome size expansion. So we annotated the transposable landscape of all of these mosquitoes. Of course, we see Aedes Anopheles always has a very small genome. They are virtually empty of transposable elements when we compare it to the non-Anopheles clade. However, we do see a massive expansion of TE content when we go down to the Aedes and Armagirus clade, particularly when we compare it to its close sister taxa of Culex. And when we specifically focus in on long terminal repeats, which are those transposable elements that are able to capture viral information, we do see that there is a very clear enrichment in Aedes aegypti, Aedes albopictus, and Armigeus for these LTRs. We also interestingly see a enrichment of this in Urinotania lowi. This is a species that, that bites both humans and amphibians. There's been a lot of work that's been done of this. We're still working this up and trying to understand sort of what its viral load is in general in the public. But this is something that our group is actively working on for Urinotania and seeing if this system has sort of evolved once or multiple times. So we now have these two sort of points. We have this non-canonical function of Peewee that's dedicating itself towards the suppression of viral targets and mosquitoes, and we see that Peewee is not functioning well for transposable elements. There's something that's allowing these transposable elements to continue to expand, even at the risk of a massive genome size expansion that we see with Aedes and with Armigeus. So our group was interested in understanding, is this just sort of a blanket feature of long terminal repeats, particularly the Tai3 family of long terminal repeats? Or are there subgroups within the Tai3 gypsy family that are capturing these sequences? That was entirely based on this image. Kept on coming back to this figure and saying, it's very strange that there's this massive bias that we're seeing in what viral sequences are being captured by these LTRs. So we took the transposable element landscape of the Orlando strain of mosquitoes that our group is working on currently and annotated it using EDTA. We took all of the Tai3 gypsy LTRs within one individual. These are all LTR families within one mosquito, generated a multiple sequence alignment, and inferred maximum likelihood phylogeny using IQ-Tree2. And so what we found is when we actually plot whether or not these Tai3 gypsy families have an Eve or not, that it actually does cluster sort of phylogenetically in this tree. And so we found that there is a small grouping of these Tai3 gypsy LTRs that are able to capture these viral sequences in the mosquito. So with that, we come back to this sort of hypothesis and whether there is a molecular domestication of these Tai3 in Aedes aegypti. And we now have these sort of 3 points of a non-canonical function that's emerged in Aedes mosquitoes with Peewee. We have Peewee not really suppressing those transposable elements, and we have a sort of clustering that seems to be indicating a sequence bias by these Tai3 gypsy LTRs. And so with that, we are continuing to work to see if there is additional modifications of the Peewee family orthologs and how this domestication has proceeded. And so with that, I want to thank you for listening to my talk. I particularly want to thank Matthew DeGennaro, my PhD advisor, Dr. Patrick Dolan at the National Institutes of Allergy and Infectious Disease, Drs. Jonna Fierce and Jason Pinar for all of their support, the entire Fierce and Pinar lab here, and all of my funders. Thank you. Happy to take any questions. Yes?
There's some older literature that seems to suggest there might be actually substantial genome size variation in between different populations or even subpopulations of Aedes albopictus.
Yes.
Do you have access to any data that might more micro-scale processes?
One of the goals that we have is to try and get a little bit more access into wild populations with long read sequences. Sadly, one of the issues in our field is most of the sequences that we have for population genomics are Illumina reads, so it's very difficult to really estimate the repeat content of these populations like that. But hopefully, fingers crossed, that's in the future. Thank you. Any other questions? All right, thank you.